everyone. So, this question I have taken uh, from the 2022 Life Sciences question paper. This is question number 74, which has appeared in the microbiology section. So, this question states that the nonsense suppressor mutation is found in options are rRNA, tRNA, start codon of mRNA, stop codon of mRNA. So let's first talk about these mutations. So there are three types of mutations. Okay. Number one, which is a missense mutation. So this missense mutation change a single codon, and so it causes the replacement of one amino acid by another amino acid in the sequence. Okay. So if I'll see there are different amino acids. So one particular amino acid is being replaced with different amino acid in the protein sequence. Okay, so that is such a mutation that happens in the sequence. So that's why the codon is now changed, so that now it is coding for another amino acids. Another mutation is the nonsense mutation. So nonsense mutation is a codon is changed to another codon, that is a termination codon or a stop codon. A stop codon. Okay, so that means if there is one amino acid chain which is being synthesized, and in this amino or in one particular codon such a mutation happens, that now the protein synthesis stops at this point only. Full length protein is not synthesized because of the presence of termination codon in between due to the nonsense mutation. The third mutation is the suppressor mutation. Suppressor mutation is usually a gene coding. It is another mutation. Apart from these two, this is a second mutation which takes place in the sequence that alters the outcome in such a way that it restores the original uh, phenotype or the original sequence. Okay. So in this topic, in this question, they have talked about a nonsense suppressor mutation. So nonsense mutations are that generates termination codon in the coding region of a gene. It causes premature termination of protein synthesis. So these nonsense mutations can be suppressed by another mutation that can read the termination codon as sense codon, restoring the synthesis of active gene product. So we can discuss about it. So for example, we have in our sequence, we have U, A, C. So this codon codes for tyrosine. Okay, and the tRNA that reads this codon that harbors an anticodon, which will be okay. So this will be the tRNA for tyrosine. Okay, and it will produce a tyrosine in the amino acid sequence. Okay, but if this there would be a mutation in this codon at this place, suppose it will become, so now this is a stop codon. It doesn't code for any amino acid. So this is a nonsense mutation. So this is a simple nonsense mutation. Now the protein sequence will be terminated. Okay, but this is the first mutation. As I have already said that nonsense suppressor mutation would be a second mutation. Would be a second mutation which takes place in the tRNA. So which changes the anticodon from this to another so this G is now altered by C, and now this can recognize, or it can bind, this anticodon can bind to the altered stop codon. Okay, so this is the mutated tRNA. Mutated tRNA, which now can code for the stop codon, and as it is a mutated tRNA of tyrosine, it will restore the wild type protein synthesis where tyrosine will be involved. So this is a nonsense suppressor mutation. So as in this example, it restored it exactly as wild type. Also, what can be done? What happens if this is another example with another amino acid or another codon that codes for lysine? Okay, so this gets recognized by the anticodon or the tRNA of lysine, which harbors the anticodon. Okay, now this undergoes the first mutation, that is the nonsense mutation. So now it becomes UAG, which is a stop codon. Okay, so now this cannot recognize this codon sequence, this tRNA. So now another mutation which is taking place in tRNA of tyrosine, which has the sequence of AUG. So here the second mutation is taking place, which is converting it to mutated one from AUG to AUC at this position. So now this can recognize this sequence and work as anticodon. So this stop codon is being recognized by this tRNA, mutated tRNA of tyrosine AUC. Okay, so as in the original structure that we are getting a protein with lysine, but due to this modification or this mutation, now we'll get a protein with, in place of lysine, we'll get another amino acid, would be tyrosine. So these are the suppressor nonsense suppressor mutation example. So it can suppress the first mutation and can restore the wild type or similar to wild type gene outcome. Okay, So for this question, so the nonsense suppressor mutation is found in, so out of the 4 example which has rRNA, tRNA, start codon of mRNA, stop codon of mRNA, will have, the correct option would be tRNA. So the nonsense suppressor mutation is found in tRNA. So this is the answer of this question. This is my NPTEL reference. Thank you.